crossing because it involves diagonal crossing. Punnett square method. How do you draw a box? You draw a box. When you draw a box, then you write. You will bring out the gamete. Capital R. You cross. You draw a line. Small capital Y. You draw a line. Okay, let me do it this way. Let me do it this way. Let us bring out the gamete. Let us bring out the gamete. You have seen capital R circle, small y, capital R circle, capital Y circle, small r, you circle it, small y, you circle it. Let us bring out the gamete. The gamete for round yellow now. Round yellow. Are you following? Am I speaking sense? And wrinkle green. The gamete for this will be, you will have RY, capital R and capital Y. Put it together, circle. That's one gamete. I have to say, the character is moving together because I love segregation. And small r, capital Y, another gamete. Am I speaking sense? So your F1 will be capital R, small r, capital Y, small y, meaning that your F1 will be round yellow. If you cross wrinkle yellow and if you cross round yellow and wrinkle green, your F1 will be round yellow, heterozygous. Am I speaking sense? Fantastic. Now, from this our F1, we will get our gamete out. So the first gamete will be RY, draw a line inside your Punnett square, capital R, capital Y, draw a line. The second gamete will be capital R, small y, capital R, small y. The third gamete will be small r, capital Y, and the fourth gamete will be small r, small y. Am I speaking sense? Fantastic. Now, you have done this one on the lateral line. You come to the other side, you do the same. Other side, you do the same. RY again, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Do you what I'm doing? Do you understand what I'm doing? Do you understand my demonstration? Do you understand my demonstration? That is fine. Let me check if I have questions. I have no question. So on the, it's like you draw X and it's like you draw X and Y axis. You draw X and Y axis. Y axis, on Y axis, you draw on X axis, you write, you put your gamete. So then you do the crossing now. In the first box, you have capital R, capital Y, small y. Am I speaking sense? Now you do the second box. Before the second box, let us do it vertically. The first line, the first vertical line. The second one will be capital R, capital Y. No, this one will be, the first one will be capital R, capital Y, capital Y. That is the first one. Capital R, capital Y, capital Y. The second one will be capital R, capital Y, small y. The third one will be capital R, small r, capital Y, capital Y. And the last one on that line will be capital R, capital Y, small y. Am I speaking sense? Am I speaking sense? Fine. So this is how you will do the crossing. At the end of the day, you will have 16 observations. What do I say? You will have 16 observations. So I will ask you to complete this assignment. I'm giving you this was an assignment. You complete the habit crossing, and you give me the phenotypic ratio, and you give me the genotypic ratio. Am I speaking sense? Am I speaking sense? Am I speaking sense? That's fantastic. So having said that, I have told you what made Mendelian experiment successful. I said he was a good bookkeeper. He studied the mistakes of the previous researcher before him. He worked on pea plant. He worked on simple pea plant with simple phenotypic ratio, with simple phenotypic characters. They're not complex traits, they're simple traits. He is of pollination of pea plant and he is of fertilization of pea plants. And there are many other people who further discover Mendel's work. Many people discover Mendel's work. So that confirm Mendel, the title they used to refer to him as father of modern genetics. Father of modern genetics. Am I speaking sense? Am I speaking sense? Am I speaking sense? For instance, like Pearson, like Watson and Crick, you know, they found discovery that corroborated. Watson and Crick are two individuals, James Watson and Francis Crick. They're the one that found the DNA structure. They're the one that found the DNA structure. So their findings further validate what Mendel did. Am I speaking sense? Am I speaking sense? Am I speaking sense? So despite one thing I want you to take away from the life of Mendel is despite the fact that Mendel was having troubles doing his experiment, he didn't give up. He kept probing, he kept fighting, he kept high level of resilience until he found what is making a green plant to give rise to a green plant. And he called that thing at that time a factor. And later, they regarded it as gene. Am I speaking sense? Am I speaking sense? Am I speaking sense? So having said that is the brief history of how study of genetics began. And that is why Mendel is called father of modern genetics. So the next thing we're going to speak to, I'll be speaking to, is I want to speak to another topic. You have known what gene is. How do gene influence our character? Gene influence every character in an individual. Height, color, shape, egg production. For instance, in animal, egg production, meat production, everything. Gene controls them. And gene doesn't do it alone. It does it with the help of environment. Environment plays a significant role in gene expression. You may be genetically brilliant. If you don't read, you'll be dumb. Am I speaking sense? Am I speaking sense? Am I speaking? If you are genetically brilliant, if you don't go to school, if you don't attend classes, if you don't do your tests, you will fail. So you need to read to bring out the best out of you. The best in you is the gene in you. So your ability to read is the environment that will help you to bring the best out of you. If you are beautiful, if you don't take your bath, if you don't comb your hair, if you don't wear good clothes, if you don't wear any clothes, you'll be ugly. So gene and environment works together to aid gene expression. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? And that will lead me to genotype by environment interaction. Genotype by environment interaction. That's what I'll be speaking to now. Genotype by environment interaction. Meaning that for you to have a very good gene expression level, environment plays critical role. What do I say? Environment plays critical role. If you have genetic advantage for growth, but if you are not eating good food, you will not look well. So you need to eat good food to help your gene for growth to express itself. Genotype by environment interaction. So if a gene is placed in wrong environment, it will not do well. A gene does well when it is placed in appropriate environment. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? Now, if you have your animals, you bought good animals from UK or from London or from overseas, you didn't provide enabling environment for them to grow, they will not do well. So their gene will not perform well. Their gene will not perform well. So taking care of them, giving them water, giving them vaccination is the environment you are providing to help that gene manifest itself. Am I speaking sense? Am I speaking sense? And this is what we call the concept of genotype by environment interaction. Concept of genotype by environment interaction. Some may say genotype gene by environment interaction, but it's not only gene. Because when the two genes coming from the father, remember law of segregation, that the character coming from the mother is controlled by two pairs of gene, and you call it allele. What's an allele? An allele is the two pairs of gene controlling the character. If the allele is dominant in homozygous form, then if you call it homozygous, it will be homozygous for that character. If the allele is heterozygous, meaning that the two characters are together, yellow, capital T and small t are together, that is heterozygous, tall. Or if the allele is capital T, it is homozygous, short. You understand? So this genotype can fall in environment. If the environment is enabling, the animal will do well. If the environment is not enabling, the animal will not do it. That is why you need an enabling environment for your animals to perform. And what is environment? What is gene? What is genotype? Genotype is the genetic constitution or genetic makeup from the two parents. And these two residing within the offspring or progeny. And what's the environment? Environment where the offspring is growing. And what's the environment? Everything outside the control of gene is called environment. Everything outside the control of gene is called environment. And everything within the control of genotype, under genotype, control of genes is called genotype. When the two genes coming from the two parents will form a genotype, genetic constitution, genetic makeup. In biology, we were taught in secondary school. Now, you have known what a genotype is, an environment is. So you need the enabling environment for the genotype to grow, to manifest. Genotype is the combination of gene from the two parents. For instance, you have a dairy cattle that you are a dairy cattle for milk production. You need, even if your dairy cattle is good for if your dairy cattle has been selected for better milk yield, if you don't give it appropriate feeding, it will not do well. So you need appropriate feeding, like provision of water, green grasses, concentrate for you to have quality milk. That's the environment. Everything outside the control of the environment is the control of this environment, like water, medication. I'm telling you components of the environment now. What am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. I'm seeing miracle. I'm seeing yeah. Fantastic. So anything within component of environment is medication, vaccination, feeding, nutrition, water, cleaning, temperature, altitude, abiotic factors, and biotic factors of the atmosphere, they fall under environment. Maternal inheritance, maternal effect. Let me explain maternal effect for you. If you go to impregnate 12-year-old girl, and that 12-year-old girl, you call it a child wife or a child mother or child pregnant girl, and somebody impregnate 35-year-old lady, they are both pregnant. The anatomy of 12-year-old girl will not provide enabling uterine environment for baby to grow, like for instance, like small uterine space, meaning the womb. And the 35-year-old lady has bigger uterine environment. You understand? So the baby cannot go beyond the size of the stomach of the uterus. So that limits the size of the baby. When both of them will give birth, the size will be different. Maternal effect. That is the environment of the uterine environment influence the growth of the baby. Nutrition, the kind of food a woman is eating when she's pregnant, influence the size of the baby, the health of the baby. That is why it is not good to marry children, because the uterine environment are not conducive for baby to grow properly. Am I speaking sense? So that is maternal influence. Maternal influence. All falls under the environment. Now, so you pick, as I'm speaking to you, phenotypic expression. Now is outward visible. What is phenotype? Outward visible expression you see in an individual. And what is the component of this? P is equal to genotype plus environment. Because genotype is an environment. Environment A, the expression of the genotype. So this leads us to variation because of differences in environment, because of differences in genetic makeup. There will be differences in the performance of your animals of an individual. That brings about variation. That's why you see in your class, some people are tall, some people are short, some people are slim, some people are fat, some people are brilliant, or not too brilliant. Some people have different characters because they have different genetic makeup and different environment. So the variation in the population is measured in terms of variance. That variance you were taught in secondary school, in mathematics, in secondary, in primary school. You use variance to measure variation in the population. Now, what we call population mean and sample mean. So when you let's say, for instance, entire Fulafia, they select students of 300 level studying this animal breed and genetics. They want to study their performance. So the entire number of students in studying animal breed of offering animal breed and genetic course become a sample from a bigger population. So we will now find the mean of your own performance and compare with the mean of the population. So you call it the mean of population universal mean, and the mean of the sample is called sample mean. Then the variation you see, you can use to interpret that population, that variation, that population. It can be form of standard deviation, and it can be form of variation, variance. So what brings about this variation in population? Don't forget, you have known what is gene. You have known how to study gene, how study of gene came about. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? Is the class interesting? If you tell, let me see your hands up. Fantastic. Now, fantastic. Now, we want to study we have known what is genotype, we have known what is environment. Your genotype of your animal need enabling environment for it to manifest itself. So the variation you are seeing in your population, your animal, is governed by genotype and environment. There are two principal component of variance. Component of variance means the next topic we'll go to is component of variance. I've spoken about variation. You understand? And variance in mathematics, in statistics, variance is summation. It says variance S square is equal to summation open bracket X, which is for the sample mean minus universal mean over n minus 1. If you remember your variance in mathematics, in statistics, am I speaking sense? What you were taught then, you cannot see the application. So that's how to measure variance. And when you measure the variance, it tells you the variation in the performance of the population of animals or even human beings. So this takes us to the next topic called component of variance. What are the components causing this variation? Number one, meaning that in your phenotypic variation, what's a phenotypic variation? Phenotypic variation is the variation in the performance of your animal within your population. Some are tall, some are short, some are tall, some are short, some are robust, some are slim, some are fair, some are dark. Am I speaking sense? Am I speaking sense? That is variation. So what are the things causing this variation? So that is phenotypic variation. Variation differences in performance within your population. And what is phenotypic variation? I want somebody to write something down. Phenotypic variation is the variation in population due to phenotype. Due to phenotype. We have phenotypic variation. Phenotypic variation is the variation in the population. Oh, let me come again. Phenotypic variation is caused by genotypic variation and environmental variation. Meaning that VP is equal to VG plus VE. Am I speaking sense? I'm coming again. Phenotypic variation is a variation in the population that is due to genotypic variation and environmental variation. Am I speaking sense? Am I speaking sense? If I'm not speaking sense, thumb down for me. Let me see. If I'm not speaking sense, thumb down for me. Okay, I'm speaking sense. So now, VP is phenotypic variation is equal to VG genotypic variation plus VE is equal to environmental variation. Don't forget, phenotype is equal to genotype plus environment. What you see in an individual, outward visible expression is the combination of the genotype of that individual and the environment where it's growing. Am I speaking sense? Am I speaking sense? Am I speaking sense? I'm seeing Abel, I'm seeing Amina, I'm seeing Charity. Fantastic. So now, phenotypic variation is a variation due to genotypic variation and environmental variation. And what is genotypic variation? Genotypic variation is a variation you see in the population that is due to genotype or due to gene action, effect of gene. Variation in the population ascribable or that is due to genotype or gene effect of gene. While environmental variation is the variation in the population that is ascribable or that is due to effect of the environment. The cleanness of the environment, the feeding, the vaccination, the maternal effect, the temperature, sunshine, rainfall, humidity. Am I speaking sense? Now, I'm speaking to component of variance. Within genotypic variation, we have other component of variation that is causing phenotypic variation. Now you write, you write VG, let me say VG is equal to VD plus VA plus VI. What is the meaning? That variation due to genotype is caused by sub-component of variance. Dominant variance plus, which is VD, plus additive variance, which is VA, plus epistatic variance, which is VI. What is dominant variance? Variation due to dominant gene effect, dominant gene action. For instance, I told you earlier, I said when you have heterozygous individual character, for instance, let me say heterozygous tall or heterozygous tall, capital T, small t, or heterozygous yellow in terms of pay plant. We are talking about pay plant. Yellow, capital Y, small y, or round, heterozygous round, capital R, small r. Capital T, small t is heterozygous tall. Capital Y, small y is heterozygous yellow. Capital R, small r is heterozygous round. Meaning that they are carrying recessive gene beside them. So what is make them to be tall? Because dominant T, capital T, is masking the expression of the smaller t. Am I speaking sense? The smaller t. Am I speaking sense? That is dominant gene action. A dominant gene action is intra-allelic interaction. A dominant gene action is intra-allelic. And an allele is a position where gene is residing on the chromosome. I told you two characters control the gene. So on that locus, two characters are there. One can be dominant, two can be dominant, capital T, homozygous tall. Can be heterozygous, capital T, small t. Then that's how you see dominant gene action. The capital T is dominating the small t, recessive T. It prevents it from masking it, preventing it from showing itself. That's the dominant gene action. That is so when you see your population of animals is heterozygous. It's a dominant gene action that makes it to be heterozygous. If the variation is due to dominant gene action, am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. So now another variation is additive variance. Additive variance is the variation in the population due to additive gene action. Additive gene action. And what is an additive gene action? Additive variance is the variation in the population due to additive gene action. And what is additive gene action? Additive gene action is additive gene action is ability of a particular animal or individual to break through, to breed through, meaning that additive gene action controls breeding value. Controls breeding value. So additive gene action is ability of an individual to breed through. I need to say ability of an individual to transfer a gene directly onto the offspring. We have two parents. They are both tall, or they are they are they are they are both fair. They are both fair. So when the two parents additive gene action disobey simple Mendelian inheritance, simple Mendelian principle, no segregation. The two genes move from the parent into the offspring in unison, jointedly. So they express themselves equally. When a gene that moves from the parent to the offspring express themselves equally, express themselves in unison equally, then additive gene action is responsible for it. Am I speaking sense? Am I speaking sense? Am I speaking sense? Additive variance is governed by additive gene action, which is ability of an individual to breed through. The character will defy all other gene action. It will move from two parents. It will disobey Mendelian principle. It will disobey law of segregation. It will disobey law of independent assortment. They will move together into the offsprings and express themselves equally. And that is the measure of resemblance. So additive gene action is a measure of resemblance in an individual. When you see that the parent, offspring look exactly like their parent, if you see the mother, you will be seeing the resemblance in the son or the children. You see the father, you'll be confused. You look like the father, you look like the mother. That the resemblance of the two parents is. If you see that, it is as a result of additive gene action. Am I speaking sense? Am I speaking sense? And that is what is called breeding value. Additive variance is mediated by additive gene action. Additive gene action mediates breeding value, ability of an individual to breed through. Am I speaking sense? So you see now that when you see variation due to genotype, which is genotypic variation, there are three components of variance that can cause it: dominant variance, additive variance, and epistatic variance. And let me explain epistatic variance from what epistasis. Epistasis is a form of gene action. Additive variance is a form of gene action. Dominant gene action is a form of gene action. Now, epistatic gene action is when a gene at one locus marks the expression of another gene at other locus. That is epistatic gene action. A gene in Maraba jumped and went to mask the expression of another gene at OMB1. That is epistatic gene action. And epistatic gene action is called interallelic interaction. Inter, between two alleles. It's not within the same allele. It jumps and goes to another location, locus. Locus is a Greek language meaning location. From locus, we get the term location in English. Locus means location. A gene in one location goes to mask, to suppress the expression of another gene at another location. It's called epistatic gene action. Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. So you see different gene action now coming together. Any of them can contribute to genotypic variance. It can be additive variance. It can be epistatic variance. It can be genotypic. It can be dominant gene action. Dominant variance. So later in the course of this study class, I'm going to teach you epistasis. That's a topic on its own. So you can now see the first layer of component of variance. Phenotypic variance is equal to genotypic variance and environmental variance. And you pick environmental variance. You pick genotypic variance, you see dominant variance, additive variance, and epistatic variance. It's not only stopping there. Now, there is something. Let us follow this equation. VP is equal to VG plus VE plus VGE plus 2 of GE. This equation is equation detailing component of variance. VP is phenotypic variance, variation you see in your population of animal or population of individual. And it's a result of genotypic variance plus environmental variance. And VGE, interaction. The genotypic variance and environmental variance used to interact together to create another form of variance, which is called VGE, genotype and environment interaction, variation due to genotype and environment. And 2 curve GE, that is a constant value, meaning that we have 2. That curve means covariation. That is, we have 2 factors that is causing variation in that population. That's one 2 curve GE, 2 covariation of genotype and environment. That's what that topic is saying, that equation is saying. And you define what this terms is, and you come and you bring out VG, and you define VG. VG is equal to VD plus VA plus VI. Am I speaking sense? 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 How many of us are in this class? We're 34. Is okay. So let me see if somebody has questions. Let me ask questions. Let me see if you have questions. Go to question and answer and ask me questions. I don't have questions yet. I don't have questions yet. I don't have questions yet. Having done this, we move to, and don't forget, there is a genetic parameter within additive gene action. Don't forget, I told you additive gene action is one that mediates additive variance. And additive gene action mediates breeding value, ability of an individual to breed through, meaning that ability of an individual to produce identical copy of itself without being affected by other form of gene action. And additive is a breeding value measure resemblance. Now, ability of a parent to transfer his character to his offspring is governed by some factors. We have heredity and heritability. Heredity has to do with how gene moves from parent to the offspring. Heredity. And when we say hereditary, means the gene has tendency to move. The gene has high potential, propensity to move. A gene that can move from generation to generation is hereditary. So for a character to be able to move from one generation of the parent to the another, that character must be hereditary. So, am I speaking sense? So that is heredity and hereditary. And it is different from heritability. Heritability is ability of a gene, ability of offspring to inherit a character from his parent. And this heritability is a measure of degree. The degree, heritability does not only speak to the gene being heritable, moving from the parent to the offspring. It has degree of expression. That's what makes it different from heredity or hereditary. Now, breeding value, which is mediated by additive gene action, is mediated again by heritability. To what extent, to what degree will a character that moves from the parent to the offspring express itself? Is it 10%? Is it 20%? That gene expression is it 40%? So a character can have low heritability, can have medium heritability, and can have higher heritability. If an heritability is measured in degree, 1%, to 100%, which is 0.1 to 1. Am I speaking sense? Am I speaking sense? Am I speaking sense? Abel said I'm speaking sense. Miracle said I'm speaking sense. Anthony said I'm speaking sense. Fantastic. That is fine. So it's either 100, 1 to 100% or 0.1 to 1. It measures degree. Now, I've told you that heritability is ability of a gene, of a character, of or an individual or offspring to have the character transferred from the parent to the offspring. That is heritability. How heritable, how can the gene move? And it dwells more on extent to which it expresses itself. When heritability is from, let me say, from 1 to 17%, is very low heritability. From 17% to 29, from less than, let me say, less than or equal to 17% is very low heritability. Very low heritability. From 17, let me say less than 29% is low, less than between less than 29% is low heritability. 29 to 40, from 29% to 40% is low, to 49% is low heritability. Heritability is low. We have from 50 to about 60%. From, let me say, look, from 40 to 60% is high heritability. So 50 to 70% is high heritability. Greater than or equal to 70% is very high heritability. Let me come again. I'm trying to come again. From, let me say, less than 29% is very low heritability. Very low heritability. 29 to 40 to 49 is low heritability. 50 to 70 is high heritability. Greater than or equal to 70 is very high heritability. And what is the implication of this? How do I explain this to you? When a gene has high heritability, you don't need selection. You don't need selection because it's not been affected by environment. A gene that has high heritability is not affected by environment. You don't need selection. So what do I mean by selection? Selection is when you have population of an animal, and maybe in January you want to rear your chicken, your cattle for Christmas or for Salah. You have seen that you pick the best, phenotypically, the big one, you select them and you feed them for Christmas or New Year. That is selection. Because all of them are not going the same way due to variation. Variation which can be due to genotypic variation and environmental variation. Because of there is variation in your population, they will not perform the same way. You would need selection. Am I speaking sense? Am I speaking sense? Am I speaking sense? That is why you need selection. Now, you need selection where effect of environment is more on that character. Because you select animal in the environment where they perform best. So animal with low heritability for certain trait, you select them. For instance, now some animal will do well in the north. They will do better in the south than in the north. While some will do well in the north than in the south. Environment. For instance, crop does well in the south than north because of fertility of the soil and availability of water. environment. So if you are planting a water-loving plant, you plant it in the south, you select it in the south. You will not come to the north and select it. Am I speaking sense? Am I speaking sense? Am I speaking sense? So a character with low heritability needs selection. A character with high heritability does not need selection. Do you know why? Because it's high breeding value. The breeding value is so high that the character can move from the parent to the offspring without the effect of environment. And we have two types of heritability. Heritability in narrow sense, which is capital HB, capital H, subscript, capital B, and heritability in broad sense. Capital H, subscript, capital B, that is heritability in broad sense. And we have heritability in narrow sense, that is H square, capital small letter H square, heritability in narrow sense. Though at times you may see some materials, some people they will denote heritability just by H square. It is still correct. But if you are going to the nitty-gritty, precision heritability, HB is heritability in broad sense, H square is heritability in narrow sense. What is heritability in broad sense? Heritability in broad sense is heritability that is, how can I say, heritability, it measures the degree of its heritability, the measure of the degree of genetic determination. How gene determine that character, not environment. That's heritability. Heritability in broad sense is a measure of degree of genetic determination in a population. And these can be caused by dominant gene action, epistatic gene action, and additive gene action. And so HB is equal to BG, variation due genetic genotypic variance over phenotypic variance, VP. That is how you measure the formula for heritability in broad sense. VG, which is genotypic variance over phenotypic variance. I have to say, heritability is a degree of genetic determination in the population. In this phenotypic variation I'm seeing in this my population, the variation that is due to genetic action alone, gene effect alone, is called heritability, excluding the effect of environment. Am I speaking sense? 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 That is HB. And then you can further, you can say it's equal to, you define what HB is. You can say it's equal to HB, VG is equal to VD plus VA plus VI over VP. That is heritability in broad sense. And what is heritability in narrow sense? Heritability in narrow sense is a measure of degree of resemblance, while heritability in broad sense is a measure of degree of genetic determination in a population. While heritability in narrow sense, H square, small letter H square is degree of measure of resemblance, which is mediated by breeding value. Which is mediated by what? By breeding value. And what causes breeding value? Additive gene action. Meaning that the variation you will see that is due to heritability in narrow sense is caused by additive gene action. So the variation you see due to additive gene action, VA, result in heritability in narrow sense. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. Fantastic. So that is the heritability. And what is the formula? We want to calculate it. I may give you some parameters and I say find heritability in broad sense or find heritability in narrow sense. The formula for heritability in narrow sense is H squared is equal to VA over VP, meaning that additive variance over phenotypic variance. Additive variance is mediated by additive gene action, which further mediate breeding value. So heritability in narrow sense is governed by breeding value. That's why you call it VA over VP. Am I speaking sense? Am I speaking sense? Am I speaking sense? So you will see now, when you say heredity, you are still talking about an entirety of the genetic makeup, gene, entirety of the gene. Heredity and environment means that genotype and environment, and what it means. But what is heredity? But what is hereditary? And what when a trait is heritable? When a trait is heritable, that trait can move from parent to offspring with high expression. High expression, that's heritable. If it is highly heritable, you don't need selection because it will just move. Environment will not affect it. Environment does not affect highly heritable characters. Only low heritable characters the environment affect. So the characters, for instance, if you do a calculation, at the end of the day, you get 29% heritability, which is low. What is the implication? You must select that environment. You must select that character in that environment where it will perform better. But if you have an 80% heritability, you don't need selection because it does not need environment to manifest itself. The effect of environment is small. Let me put it that way. Because we cannot rule out the environment Gabadaya. The effect of environment is very small. The effect of environment cannot really alter its expression. Am I speaking sense? Why a gene that has low heritability has a pronounced effect of environment in the expression. A gene that has higher heritability has low effect of environment in the expression, manifestation of the expression. Am I speaking sense? Let me give you certain value. For example, sheep and goat. Litter size. Litter size means the number of animals a goat will give birth to at a go. A doe, a buck. A doe will give birth at a time. If it is sheep, the number of piglet, of litter, the number of offsprings, and he will give birth at a time. That's litter size. In pig, the number of piglet a swine will give birth to at a time is called litter size. The litter size in goat is 10% heritability. So you need selection. Then, if you need selection, effect of environment is high. Then you must make the environment enabling, providing water, providing feed, providing medication, vaccination, spacing, avoid overcrowding. So you use the environment to show up, to increase the performance of the animal because the character has low heritability. Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. Birth weight. I told you the other time, a 12-year-old girl that got pregnant and 35-year-old lady that got pregnant, the size of their baby will not be the same because there is correlation between the uterine environment, the size of the uterus, and the size of the baby. A child of 12 years with pregnancy has small womb, uterus, and the baby cannot grow beyond the size of that uterus. And the lady of 35 years has bigger uterus. We will give environment for the baby to keep growing. Am I speaking sense? Am I speaking sense? That is why don't go and marry children, babies. They're endangering their life and the life of the baby because their physiology, though they have breasts and all that, they are not physiologically mature. Anatomic growth is different from physiological growth. Anatomical growth is a girl began to show breast, began to show hair on the armpit, does not mean physiologically, the baby is mature to be put in the family line. Am I speaking sense? Am I speaking sense? So you look at birth weight. So the size of the mother determines the size of the baby. That correlation between the size of the mother and size of the baby. So the birth weight is 40% heritability. Is low. It's not, but it's low. You need environment to increase it by giving feeding, vaccination, medication, and all of that. Milk yield. Milk yield has 40% heritability or thereabouts. So you need environment by giving feed. In fact, green grasses increases the quality of milk than giving concentrate. Survival. When you want survival rate of your win at win. I'm speaking to sheep and goat here. Let me move to cattle. The weight gain in cattle is 65% heritability, is high. So you just need little feeding, medication, just provide an enabling environment. The cattle, because genetically cattle is meant to grow big. That is what it means. You understand? So we go to poultry. You go to poultry, egg weight is 50%. Body weight is 55% heritability. Egg production is 10%. Meaning that you must feed your chicken very well. You must provide them water. You must provide them vaccination. You must provide them medication for environment, for you to have more egg production. Low heritability provides for selection and provides for you to provide more enabling environment for them to grow and perform. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? So in the next class, I'm going to teach you gene linkage, type of gene action, gene action. Then from there, I'll teach you gene linkage, and I'll teach you epistasis. After this class, I want the children to talk to me. I want to ask them some questions. So your next class will be, I'll speak with your HOC and we'll get across to you at least a day to the class. I'll speak to your HOC, your head of class. Let me see. Ask me questions. Let me see if you have questions. Nobody's asking question. Okay, I'm seeing question here. I'm coming. Let me answer this question. I'm seeing question here. I'm seeing question here. I'm coming. I'm seeing question here. Okay, I don't know. Let the person that asks because let him send his name or her name. The question is, does a mother get trait from the father after giving birth? One question, does a mother get trait from the father after giving birth? Is there a transfer of gene from parent to parent? No. There's no transfer of gene from father to father. The gene is moving from the father laterally and from the mother laterally into the offsprings. They are the one contributing their gene to producing the offsprings. If it's the character that is largely controlled by Mendelian inheritance, law of segregation will happen, independent assortment will happen. So both of them are giver. They are the one producing the children. So the gene cannot move. Rather, the gene move from the father to these children, from the mother to the children. Another question is, does the mother get traits from the father after giving birth? No, you don't. Contribution. Contribution contribution to production of children, offspring, or progeny is contribution from the two parents. It's laterally moving into the children, not cross-laterally between the father and mother. No. I told you earlier, every character, higher animals, mammals, every character is diploid. We are diploid organism. That is why a man has 23 pair of chromosomes, a woman has 23 pair of chromosomes. So during gametogenesis, during reproduction, the 46, man has 46. 46. The woman has 46. During gametogenesis, the mother will give 23 out of his own 46. The man will give 43 out of his own 46. And the offspring will have 46. They contribute equally, except where there is mutation and some other factors. Am I speaking sense? Am I speaking sense? Am I speaking sense? Another question is, ask me questions. I want to ask somebody's asking me another question. If that question, if my response, my answer makes sense to you, let me see your hands. Let me see your thumbs up. Fantastic. So you and your boyfriend, let me say Aisha and Joanna, they cannot exchange character gene between each other. They can only contribute the gene to the offspring. The gene coming from them is coming in haploid nature, and it will undergo segregation. 2N will form N haploid. And by law of independent, that is law of segregation, law of independent assortment, the N will assort independently, the N will combine independently without asking each other questions randomly. That is what gives rise to diversity, variability. No mago, they assort independently, they combine independently of each other. That is what it means. And you will now see the manifestation in the offspring. Am I speaking sense? Another question is, does a mother get traits? I've answered this one. Sir, can the child transfer gene gotten from the father to the mother? No. The child cannot transfer the gene obtained from the mother to the father. Like I told you, is a directional movement. The gene is coming from the two parents in one-way direction, in not a dual direction. Is the one-way direction from the parent to the offspring. It will not go back. It will not go back. That is why they contribute to produce the offspring. Let me explain something to you. Immediately, Johanna sleeps with Aisha. The sperm enters the egg. That's what we call syngamy. Syngamy will happen immediately. Syngamy paves way for fertilization. Syngamy is when the fusion of the nucleus of the sperm with the sperm of the egg. When that one happens immediately, reproduction begins. Cell division begins. We have cell division into thousands of division. We were up to maybe up to 15 level of self division. There where we have stem cell. At 16 level of self division, stem cell can form any individual. Stem cell has ability to grow up and become an individual. At above 15 level of cell development, at 15 level of cell development, the cells are totipotent. They can form individual. Pick one at that level, culture it to go to become an individual. But immediately after 16 level of cell development, the cell begin to determine their fate. FATE. They begin to give them assignment. They become multipotent. Maybe this one you go and form all the cells within the brain. They can form anything within the brain. You go and form anything within, that is pluripotent, rather. They're pluripotent. Go and form all this, anything within the intestinal area. They're pluripotent. I think a pluripotent cell is a cell that can form different organs, but their fate has been reduced. A totipotent cell has undetermined fate. It can grow and form anything. But a pluripotent has a limited fate, but it can form multiple organs. Immediately, the cells are given assignment. You go and form the shape of the head. Go and form the shape of the eye. You go and form the shape of the leg. They cannot form another thing than their final fate. So during the process of forming all those characters, if mutation happens, then the eye may not be there. The shape of the egg may be somehow, and all that. That is mutation. But maybe if you come for MSc class, we can be talking about mutation. Am I speaking sense? Am I speaking sense? Am I speaking sense? Tony said I'm speaking sense. That is fine. So I'll be ending this class here. In the next class, I'll communicate to you. In the next class, I'll be taking gene action. You realize the class is entering each other. For what is a gene? How we study gene, how gene interact together, then how gene react with the environment, how we have variation in our environment, what is causing the variation, and heritability, and all of that. Isn't it a fantastic class? Isn't it a fantastic class? Tell others that the class has started. So next time, we're going to talk about gene action. I'm going to enjoy this class. I welcome you back from the new year. I wish you a very happy new year. I pray to read your books so that I can do well. I pray that this year will be peaceful and Nigeria will be peaceful again. God will protect us from bandits and terrorists. In Jesus' name. Thank you. Bye-bye.